Hello students, today we learn about bioinformatics. In this lecture, I am just overview the different type of application of the bioinformatics, what is that and where the bioinformatics can be applicable in the bioscience, in the life science, in the microbiology, in the biotechnology. So let's start. Bioinformatics, it is the combined science with the three type of the data. First one is the biology, second one is statistics, and third one is the computer science. So the bioinformatics is regarding the two words there, bioinformatics. So the biology database are in couple or they have a get together to the IT, that is the information technology. So that is known as the bioinformatics. In the bioinformatics, the three things can be the important: the biology related to organisms, statistics, different type of methods are there, and the computer science. So without computer we can't be interrupts or involve the biological databases, and without statistics we can't be calculate all these things. So the bioinformatics is that. Other way, the data science, that is the knowledge of the domains, statistics and computer science. So in the data science, only the different type of data can be the egg, rival, and they have to study. But in the bioinformatics, the data is biological data. These are the living organisms data, can be the interrupt, can be the study out with the science, and that is known as the bioinformatics. Life, ATGC. So the ATGC is nothing, it is the DNA sequence. So our life is only regarding to the ATGC. So the human beings sequence is the ATGC, the bacillus sequence is the ATGC, E. coli is the ATGC, and any type of the organisms which is the couple of this is the combined of ATGC sequences of the life. So this one ATGC is known as a biological database. So that biological database are the two types, the nucleotide databases and the protein databases, and from that databases we can study out the different type of logical and scientific areas. These are the databases. So the different type of databases are there. The first one is genome database, sequence database, structured database, microarray database, chemical databases, metabolic databases, enzyme databases, disease databases, literature database, taxonomy databases. So all the type of databases are related to the organisms, and they have the basis of the types. So if that all databases are included in the major category, so that is known as the two type of the databases. One is the primary database and second one is the secondary database. So these are a source of the databases on the basis of the source. And the second one is, if we got the special categories, so integrate databases and the composite databases. So these are the biological databases are included in the bioinformatics science. So these are the flow charts. Which type of databases are known as the biologically? So the first one is structured databases and include the PDB, SCOP, CATCH. These are some site or some databases are there. Second one is the sequence databases. That sequence database is again divided into the two types, the protein and nucleotide databases. In the protein, the Swiss Prot, PIR, TRIS, EMBL, PROCIT, PFrame are included in the protein. And in the nucleotide acid databases, the gene bank, DDBJ, EMBL, these are some biological databases are included. And the last one is the other type of the databases, just like the enzyme databases, disease databases, literature databases, metabolic databases. So these are a different type of categorized into the databases. So the major type is the three: structure, sequence and other. Or in the structure, the protein databases are there, the structure of the proteins are there. Or in the sequence, these are again divided into the two types, protein and nucleotide databases. This one is the dot plot techniques. That techniques is summarized, and that is for a sequence alignment. Which type of sequences is the similar alignment, and from the dot matrix or dot plots we can know the flow of energies. This one is a application of the bioinformatics. Second one is the application is known as the alignments. So these alignments are the two types, local alignments and global alignments. From this alignment we can sequence the similar sequencing, and from that alignment we can study out, we can search out the organisms are how far to the other, the ancestors and the relations between of the two organisms can be found out from this technique. So this one is the DNA to messenger RNA. We know about this technique, so we include that technique to study out. Messenger RNA to protein, we know all these things, how the messenger RNA become the protein from the genetic code, so we can output or benefit of these techniques. Third one is the genetic codes. So we know different type of genetic codes are there. One amino acids have 3 to 4 genetic codes or amino acid sequences. So couple of that all sequences in the load with the software of the bioinformatics, and we can just convert the DNA to the messenger RNA to the proteins, and that proteins can be the study out with the structure analysis. So next one is known as the gene predictions. We can predict the gene from the different type of the practical or different type of the tool of bioinformatics. The gene predictions, that is again divided into two types, the protein coding and non-protein coding. So that the homologous basis gene predictions is very important. Or in the non-protein coding, the tRNA, rRNA and sRNA we can include to prediction of the gene in the whole sequence. Drug designing, this one is a very important application in the pharmaceutical. So today the pharmaceutical companies can design the drug, because that experimentally the drug is designed in 10 to 15 years, but in the bioinformatics we can 2 and 3 days design the drug, and we study all these things. So this one is a very important role of the bioinformatics in the pharmaceutical companies. This one is a flow chart for the designings. So we discuss about the later about that is a long topic, but just application regarding of the bioinformatics. This one is a very good application of the drug designing. The 3D structure of protein, we can analysis of the 3D structures, we can analysis the 3D structure of protein predictions. So from the sequence of the DNA we can just transcripts and just we synthesize the protein, and we can analyze which type of structure of protein can be done by the DNA or messenger RNA. This one is the rotations of the amino acids. So in the messenger RNA or in the proteins, some amino acids are there, and that amino acids are rotated, and that rotations can be analyzed by an Indian scientist. The name is Ramachandran, and that plot can be developed, that known as the Ramachandran plot, and from that we can study out the which amino acids can rotate in which angle. So this one is study is very important for the drug designing. Protein analysis, we can also analyze the protein with a different type of software, just like the RASMOL. This one is a view of the RASMOL. The RASMOL is the software for the bioinformatics, and we can predict or analysis of the protein. The microarray, this one is a very good techniques, and from this technique we can array the normal cells or the cancer cells, and we culture, and messenger RNA and the cDNA can be the couple up, and in the one plate we can analyze the normal cell or the cancer cell with the plating, and from that array we can just identify the which type of the gene are involved in the cancer cells. This one is the known as the microarray technique, and that very good application of the bioinformatics. This one is a view, the whole view of the microarray. That dot is indicate the different type of genes, and from this dot we can calculate the normal cells, how different to the cancer cells, and we can find out which genes are here spot in the cancer cell or not in the normal cell. So we can compare the normal cell and the cancer cells. This one is the components. The different type of the components are included, that the development of the software or the databases. So when we talking about the components of the bioinformatics, these are a major part of the three: the DNA database or the biological databases, software and tools are involved in the components of the bioinformatics. Thank you.